Usually, when we think about aesthetic treatments, filling a wrinkle or smoothing out a scar, there's this underlying expectation of basic mechanics.
Right, it's a very mechanical process.
Yeah, it feels a lot like spackling a wall. Honestly, you have a dent or a hollow, a doctor injects a gel into that space, and the dent goes away simply because the gel is physically sitting there. It's taking up room.
Exactly. It's a purely volumetric approach. The mechanism of action is literally just the occupation of space within the dermal or submucosal layers.
It's visible, it's immediate, and I mean, it makes intuitive sense to anyone who has ever patched drywall. But stepping into the world of regenerative medicine and biological age hacking, that throws that entire spackle concept completely out the window.
It really does. We are looking at a completely different paradigm today.
Right, because what if injecting fragments of fish DNA into your skin doesn't just blindly fill a space, but actually hacks your cell's biological clock to build entirely new natural tissue from the ground up?
Yeah, we're moving away from the concept of a passive inert implant. This is all about introducing an active biological catalyst, something that fundamentally changes how the surrounding tissue behaves.
Which is exactly the journey we are mapping out for you today. Welcome to the deep dive. Our mission today is to track the exact chronological timeline of polynucleotide gel matrices, these high molecular weight DNA fragments. We're tracking them from the literal second they are injected into the tissue through their biological half-life to months and even years down the line.
And we pulled a genuinely fascinating stack of sources for you to explore this with us.
We did. We're looking at clinical aesthetic guides, regenerative medicine studies, and we're even parsing through some pretty heavy biochemical texts on chemical DNA sequencing and macromolecule complexation.
That combination of sources is vital, though, because to really grasp the clinical outcomes, the visibly thicker skin or the faded scars, you have to bridge the gap and look at the raw biochemistry happening at a microscopic level.
Absolutely. But let's put the central paradox on the table right away. People casually refer to these polynucleotide injections as fillers, yet looking through the timeline data in our sources, these gels actually disappear from the body relatively quickly.
Yes, their biological half-life isn't permanent at all.
So how can a product that degrades and vanishes from the tissue so fast create structural results that last for over a year?
Well, unraveling that mystery really requires us to rewind the clock to before the needle even touches the skin. To understand the timeline of how this gel behaves and integrates inside the body, we have to look at the blueprint.
Right.
We have to know exactly what is being injected and how science guarantees its structure. Manufacturers are not just, tossing salmon into a blender and hoping for a medical-grade outcome.
I am so glad you brought that up because reading these sources, my immediate reaction was definitely skepticism. They describe these as high molecular weight DNA polymers, up to 8,000 kilodaltons, extracted mostly from the gonads of salmon or trout.
Which sounds intense.
It does. DNA is the literal blueprint of life. It is overwhelmingly complex. So how do we even know we have the precise fragments, the right chain lengths, and the exact physical structure required to make a safe, stable gel for human injection?
The assurance really comes from foundational biochemistry. Our sources point specifically to the Maxim Gilbert chemical sequencing method.
Maxim Gilbert, okay.
Developed in 1977. This is one of the very first ways scientists learned to actually read the genetic code. The chemical principles established by that method still allow manufacturers today to verify the exact sequences and structures of the DNA chains they extract.
See, sequencing DNA always sounds to me like trying to read a 1,000-page book where all the pages are microscopic, glued together, and written in a language you don't even speak. How does the Maxim Gilbert method actually parse that out so we know we have the right structural polymer?
It relies on statistical chemical modification, which is just a brilliant manipulation of molecular bonds. You take the raw DNA sample and expose it to highly specific reagents that target particular bases, the distinct letters of the DNA code.
Okay, right.
One of the primary reagents used is dimethyl sulfate. This chemical specifically targets the purines, which are the adenine and guanine bases.
Methylating them, right, meaning it attaches a methyl group directly to the molecule.
Exactly. And the placement of that methyl group is critical. It attaches to the N7 position of a guanine base, for instance. That attachment creates a positive charge that completely destabilizes the bond, linking that specific base to the DNA sugar backbone.
Oh, wow. So it just breaks it.
The base effectively pops off, leaving the sugar exposed and vulnerable. Then, by introducing alkaline conditions, The sugar phosphate backbone breaks entirely precisely at that spot.
So you are intentionally fracturing the DNA chain at very specific predetermined letters.
That is the core mechanism, yes. And they run a parallel process for the pyrimidines, the cytosine and thymine bases, using a different chemical called hydrazine. By performing these controlled partial cleavages across different samples, you generate this massive mixture of DNA fragments of varying but predictable lengths.
Okay, but how do you read that mess?
Well, to actually read the sequence, those fragments are run through polyacrylamide gel electrophoresis, which is commonly known as PAGE.
Oh, I've seen images of page results in textbooks. It looks like a glowing barcode with all these little illuminated dashes stacked in columns.
That glowing barcode is the sorting mechanism. The gel matrix in the PAGE setup sorts all those fragments strictly by size. Because the fragments in adjacent bands differ by just a single nucleotide, a single letter, you get a readable ladder. By reading straight out the ladder, scientists determine the exact sequence of the original unbroken DNA chain.
That is wild. So taking this back to the aesthetic treatment, this deep chemical understanding proves that the manufacturing process isn't a guessing game at all.
Not even a little bit.
By knowing exactly how to cleave, verify, and analyze these chains, manufacturers can precisely shear the raw fish DNA into highly purified polymers of a specific molecular weight. They strip out all the proteins, all the active genes, and like anything that could trigger an immune response. What is left is simply the pure structural polymer chain.
And those structural chains have a very specific property.
Yeah.
Because of the phosphate groups running along the DNA backbone, these chains are highly hydrophilic. They aggressively attract and bind to water.
Which is key for a gel.
Right. When these purified chains cross-link together, they form a three-dimensional porous scaffold. It becomes a highly viscous, water-binding gel matrix.
Which our sources highlight as being completely distinct from the low molecular weight liquid versions of this technology. Those are known as PDRN, right?
Exactly. PDRN is a free-flowing liquid primarily utilized for topical wound healing or joint issues. Polynucleotide, or PN, due to its high molecular weight and cross-link structure, is a thick, substantial gel specifically engineered to physically restructure dermal and submucosal tissue.
Okay, so we have our blueprint. We have a chemically verified, purified 3D scaffold of structural DNA. Let's start the clock on the timeline. Phase 1, the immediate aftermath. What physically happens the very second this viscous gel is injected into the tissue layer?
So the first 48 hours are dominated by physical presence and a dramatic shift in osmotic pressure. When that highly concentrated gel is introduced into the submucosal or dermal tissue, it completely alters the local environment.
How so?
The surrounding tissue normally sits at a resting osmotic pressure of about 280 milliosmoles per liter. The introduction of the gel spikes that pressure instantly up to 308 milliosmoles per liter.
That feels like a massive localized disruption. I mean,
Really?
the body's going to immediately try to equalize that pressure gradient, basically turn the gel into a sponge.
A very aggressive sponge.
Yeah.
Water from the surrounding tissues and capillaries rushes into the injection zone to balance the sudden pressure spike. By day 3, the dermal water content in that specific area jumps by a staggering 15%.
Wow, 15%. So for someone looking in the mirror on day 3, they're seeing deep hydration, maybe a little bit of swelling or tissue expansion, but the effect is purely mechanical and physical at this stage.
It is entirely physical volume and water weight. However, on a cellular level, the alarm bells have already rung. By that same third day, macrophages, which are the immune system's primary cleanup and assessment crew, arrive at the site to investigate this new foreign material occupying the tissue.
The sources actually provide an incredible visual for the mechanical benefit of this immediate physical phase, especially concerning the treatment of deep atrophic acne scars using a technique called subcision.
Yes, subcision is a great example here.
Right. For anyone unfamiliar, imagine a deep, depressed scar that is tethered down by rigid fibrous bands underneath the skin. A doctor inserts a needle or cannula and physically slices through those bands, releasing the tension and lifting the tissue.
Which works great initially.
But the problem is, if you just leave that freshly cut pocket alone, gravity and the body's natural healing process will often glue the roof right back down to the floor.
The readhesion rate is notoriously frustrating for doctors. The clinical data shows that without any intervention, approximately 35% of those freshly broken fibers will simply readhere to each other within 2 weeks. The scar just sinks right back down.
And this viscous gel acts exactly like construction scaffolding. If you inject the polynucleotide matrix right into that newly cut pocket, Its high viscosity physically holds the space open, it props the roof up.
Exactly.
Because it forms a dense 3D mesh, it physically blocks the severed fibroblasts from touching each other. By simply occupying that space, the retention rate drops from 35% to less than 5%.
It's a huge drop.
The scaffolding buys the body's cellular workers enough time to build a brand new floor before the roof has a chance to collapse.
That scaffolding analogy perfectly sets up what the regenerative medicine sources refer to as the biological half-life paradox.
Oh, let's dig into the paradox, because the survival timeline of this gel is wild to me.
It really is. See, the body is exceptionally good at destroying loose genetic material. Your blood plasma and cell membranes are heavily patrolled by enzymes called nucleases. Their entire job is to seek out and chop up free-floating DNA, preventing rogue genetic information from causing chaos.
Makes sense. You don't want random DNA taking over.
Right. So if you were to inject naked polynucleotide chains into the submucosal layer, just the free-floating unlinked liquid, those nucleases would obliterate it. Naked PN degrades in vivo in just 2 to 3 hours.
2 to 3 hours is barely a blip. The body clears it before it can even initiate a meaningful tissue response.
But the physical structure of the product changes the rules of engagement. Because we are injecting a high molecular weight, highly viscous cross-linked gel matrix, the nucleases face a physical barrier. They cannot instantly saturate the material.
Oh, I see.
Yeah, they can only attack the exposed surface area of the gel blob. They are forced to slowly chew their way inward from the perimeter. This mechanical resistance extends the degradation half-life of the gel from 3 hours to a window of 14 to 28 days.
That's incredible. Simply by structuring the DNA chains into a dense, interwoven mesh, you buy the tissue nearly a month of sustained exposure.
And the biochemistry sources suggest this window is about to become even more controllable. Scientists are currently experimenting with complexing these polynucleotide chains with a substance called schizophilin.
Schizophilin. That sounds like a synthetic chemical, but what actually is it?
It's actually a naturally occurring beta-glucan derived from certain fungi. When scientists wrap the DNA chains in this schizophilin complex, it acts as a molecular shield against the nuclease enzymes. The physical barrier is so effective that studies show it slows nucleose-mediated hydrolysis to 1/6 of its normal biological speed.
Wait, 1/6. By utilizing that beta-glucan wrapping, manufacturers could theoretically dial in the exact half-life of the gel.
Precisely.
They could formulate a scaffold that survives for 3 weeks or 6 weeks or 3 months, completely customizing the degradation timeline based on the specific tissue defect being treated.
It elevates the gel from a simple scaffold to a highly programmable delivery mechanism.
Okay, so following the timeline, we move past the initial swelling and into weeks 2 through 4. The gel has been acting as a prop, holding the tissue open, but those nucleases are relentlessly chewing away at the surface. The matrix is breaking down. Where does all that molecular debris go? It doesn't just evaporate.
The degradation debris is the true active ingredient of the treatment. Around day 10, the localized healing response, triggered by the physical presence of the gel, causes capillary blood flow in the area to peak.
Okay.
The flow velocity increases by 0.4 centimeters per second, flooding the tissue with oxygen and nutrients. By day 14, specific enzymes called endonucleases start making serious headway into the gel, slicing the long structural DNA chains into smaller, free-floating oligonucleotide monomers.
And those little monomers just drift out into the newly vascularized tissue?
They drift through the extracellular matrix and physically bind to highly specific receptors on the surface of your cells, specifically the adenosine A2A receptors located on the fibroblasts.
Fibroblasts being the cells that make collagen, right?
Exactly. They are the cellular factories responsible for synthesizing structural proteins like collagen and elastin. Additionally, as these monomers circulate, their actual molecular architecture provides a secondary benefit. The nitrogenous rings within the DNA bases act as molecular traps. They physically catch and scavenge free radicals, pulling oxidative stress and inflammation out of the tissue environment.
We need to pause on a crucial question here that, frankly, any listener tracking this biology is likely asking right now. We are talking about injecting fragments of fish DNA into human tissue, having it break down, and watching those fragments physically bind to human cells. Is this integrating into the human genome? Is the genetic code of the patient being altered?
It is the single most common concern patients have when introduced to this technology. And based on comprehensive clinical and biochemical data, the answer is an unequivocal no. There is zero alteration of the genetic code.
Okay, good. Walk us through the cellular barrier that prevents that.
These oligonucleotide monomers are simply building blocks. They remain entirely in the extracellular space, or the cytoplasm. They do not possess the delivery mechanisms required to penetrate the nucleus of your cells, which is the heavily fortified vault where your actual chromosomes reside.
Right, the nucleus is locked down.
Exactly. Think of your cellular DNA like a massive, secure reference library. The injected polynucleotides are the equivalent of a truckload of loose, disconnected letters being dumped in the parking lot outside. Your body recognizes the raw material. It either sweeps up those loose letters and uses them in the salvage pathway, recycling the purines and pyrimidines to build its own natural cellular energy, or it simply metabolizes them completely into uric acid, which is then harmlessly excreted by the kidneys.
The parking lot letters analogy clears that up perfectly. No books inside the library are being rewritten. The cells just salvage the raw materials. So those A2A receptors are activated by the debris, and the oxidative stress is cleared out. What does the clinical data show happening in the tissue as a result of that activation between weeks 2 and 4?
The cellular shift is explosive. By day 14, the proportion of fibroblasts entering the DNA synthesis phase, the S phase of the cell cycle, where they are actively preparing to divide and manufacture proteins, jumps from a resting baseline of about 2% all the way up to 18%.
Wait, a 9-fold increase in cellular factory activity in just 2 weeks?
Yeah, it's massive.
Yeah.
By day 21, the absolute number of proliferating fibroblasts increases by a full 25%. They're multiplying rapidly and working at maximum capacity. By week 4, these high copper-activated fibroblasts have pumped out so much new structural protein that the actual measurable density of the extracellular matrix in the dermis has increased by 10 to 15%.
The physical scaffolding has effectively transitioned into a biological rebuilding phase, which pushes our timeline into the long game months 2 to 12 and beyond. The sources highlight a fascinating milestone at day 180, or 6 months post-injection. By that point, the original gel matrix is 100% metabolized. It is completely gone, broken down into uric acid, carbon dioxide, and water, and flushed from the system.
Not a single trace of the original fish DNA remains in the tissue.
Yet the most dramatic clinical results, the smoothing of fine lines, the lifting of atrophic scars, the overall thickening of the skin, actually peak around months 2 to 3 and remain highly visible for well over a year. If the injected product is entirely absent, why does the result persist?
The regenerative medicine sources use a brilliant analogy to explain this sustained outcome.
Yeah.
It is exactly like going to the gym and lifting weights.
Okay, I love this. So the gel injection is the workout. It introduces mechanical stress, it provides the salvage pathway fuel, and it forces the fibroblast muscles to work incredibly hard.
Yes.
Eventually, the workout ends. You leave the gym, and the gel leaves your tissue. But the muscle mass you built during that workout, the new collagen and elastin fibers, that is real physical tissue. It was synthesized by your own body, it belongs to you, and it stays with you long after the stimulus is gone.
And contextualizing this within the broader landscape of aesthetic medicine, it highlights a stark physiological contrast with standard hyaluronic acid or HA fillers.
That is a critical comparison, as the two treatments are constantly confused or grouped together under the umbrella of injectables.
Right. An HA filler is largely inert biology. When injected, it occupies space and pulls in water to create volume. Over a period of 9 to 12 months, your body's naturally occurring hyaluronidase enzymes slowly degrade the HA gel until it is entirely consumed. Once the HA is gone, the artificial volume is gone, and the tissue returns to its exact baseline state. Polynucleotides, conversely, stimulate an autologous volume increase.
Autologous meaning derived from the individual's own body. It is your own natural tissue.
Exactly. The volume, the lift, and the density visible at month 6 are not the gel. It is the newly synthesized network of type I and type 3 collagen. Under normal physiological conditions, your body's type I collagen fibers are incredibly resilient. They possess a natural half-life of 10 to 15 years.
A 10 to 15-year half-life for the actual collagen fibers produced. The clinical timeline shows that between weeks 8 and 12, the synthesis of this specific new collagen reaches its absolute maximum output. For patients utilizing this to treat the deep atrophic acne scars we discussed earlier, the overall depth of the scar reduces by an astonishing 30 to 50%, driven purely by this new autologous tissue pushing up from below the dermal floor.
Furthermore, the overall health of the tissue is fundamentally upgraded. Ultrasound scans taken at the 6-month mark reveal that local blood perfusion, The complex network of microcirculation feeding that new tissue remains 25% higher than the pre-injection baseline.
Wow.
Yeah, the tissue is quite literally acting younger and receiving vastly more nutrients.
However, for anyone listening and considering the real-world application of this biology, the sources emphasize a massive caveat. This biological hacking is not a permanent magic spell. The longevity of these autologous results is inexorably tied to your daily lifestyle choices.
Because the newly synthesized collagen is biologically yours, it is subjected to the exact same environmental degradation and aging factors as the rest of your tissue. The most aggressive and destructive factor being ultraviolet radiation.
Unprotected sun exposure.
Right. Clinical studies demonstrate that regular, unprotected exposure to daily UVB radiation can shorten the lifespan of this newly built collagen network by up to 35%.
35%, that's a huge loss.
It is. The UV rays act like microscopic scissors, indiscriminately snipping and fragmenting the exact collagen fibers the fibroblasts just spent 3 months desperately building.
This explains why the visible clinical results gradually soften or appear to fade over a 6 to 12-month period without intervention. The patient isn't losing the gel has been gone since month 1. The patient is naturally degrading their own newly built collagen through daily environmental damage and the relentless march of the biological aging clock. That is exactly why the clinical protocols recommend a maintenance injection every 6 to 12 months. It provides those resting fibroblasts with another intense workout, spiking the synthesis rate back up to outpace the natural degradation.
It basically establishes a continuous proactive cycle of biological tissue upkeep.
Let's pull all of this together and summarize the incredible timeline we've just tracked. We start with a raw, natural material DNA extracted from fish gonads. Using the precise chemical sequencing principles of the Maxim-Gilbert method, scientists can verify, shear, and purify these chains into a highly specific three-dimensional gel scaffold. When injected into the tissue, it initially acts as a physical shield and a sponge, pulling in water and drastically dropping scar refusion rates.
Right, the mechanical phase.
Then, as it slowly succumbs to the body's nucleuses over a few weeks, it releases oligonucleotide monomers that lock onto A2A receptors, transforming resting fibroblasts into hyperactive collagen factories. Finally, the gel vanishes completely from the body, leaving behind a dense, newly synthesized matrix of natural human tissue that can persist for years.
It is an exceptionally elegant process. It leverages our deep understanding of macromolecule biochemistry to completely redefine what an injectable treatment is capable of achieving. But looking at the trajectory of this research, there is a final concept hidden in these sources that pushes the boundaries of regenerative medicine even further.
What's that?
Well, right now, the industry extracts these DNA fragments from fish simply because salmon provide an abundant, highly biocompatible source of raw building blocks that trigger a broad, generalized healing response. We inject it, and the tissue broadly rejuvenates. But knowing that we can map the exact sequence of DNA down to the single letter using chemical cleavage methods, and knowing that we can dictate the precise degradation timeline of a gel using schizophilin beta-glucan shielding, what happens in the near future when we abandon raw extraction entirely and start synthesizing completely custom polynucleotide sequences in a lab?
Oh, that's fascinating.
Right.
Yeah.
Will we eventually inject programmable DNA gels that don't just trigger general collagen production, but carry specific structural instructions, effectively acting as biological software updates that tell the skin exactly what shapes to form and when to dissolve?
A literal software update for your skin. That is an incredible thought to leave you on today. Thanks for joining us on this deep dive, and we'll catch you next time.